Our next lecturer is Alex Young. He's been here, I'm sure you've all met him by now. He's here the entire time. Alex got his MPhil in computational biology from Cambridge and PhD in genomic medicine and statistics from Oxford, and then did a postdoc at Decode in genetics in Iceland, which got him access to amazing founding data, which he's used to excellent effect, and then a postdoc at the Big Data Institute at Oxford. He is, in my opinion, the most brilliant young statistical geneticist out there. And when he was on the market, you're not a statistical geneticist. Patrick too, but Patrick is you said young too. Patrick's an economist also. So when Alex finished his postdoc and was on the job market, of course every great genetics department wanted to hire him. So we're very lucky that he decided to instead work with us at the SSGAC. So he's been continuing to do brilliant work that he's going to tell us about. Thanks, Alex.
Oh, thank you, Dan. I'm not sure I can live up to that introduction, but hopefully you'll learn something. So I'm going to talk about gene-environment correlation, although perhaps a more accurate title would be sort of how to use family data properly to disentangle different things. And now my clicker, this doesn't seem to be working. That's annoying.
Just click on the screen something that just needs accurate path. Or like with your with the mouse.
Oh yeah. Okay, yeah. So why do we want to use family data? And I think the most compelling reason is because standard methods that have been used in genome-wide association studies for the past 10, 15 years, they've been very good at finding SNPs and genomic loci that are reliably associated with traits, but they're not going to give you unbiased estimates of the causal effects of inheriting particular alleles. And this is because there are various factors that contribute to genotype-phenotype association in the population. This is from a review paper that we published in Science in 2019. And in the standard GWAS methodology, you're basically regressing phenotype onto genotype, potentially with some controls like principal components that do reduce confounding but don't necessarily eliminate it. So what you're picking up is the causal effect of inheriting that allele or other alleles close to it in the genome, plus what could be termed as confounding effects, which include indirect genetic effects from relatives. Whether you want to class them as confounding effects or not depends a little bit on your perspective. In some sense, they are a causal effect of the allele just going through the environment. And what I mean by that is if you inherit an allele from one of your parents, and this allele raised your educational attainment, it would have also affected your parents' educational attainment. And by affecting your parents' educational attainment, it can affect you through the environment. And that would be called an indirect genetic effect from the parents, as opposed to a direct genetic effect, which is when the allele you inherit has an effect on your own trait. And they could also go through other relatives, such as siblings. And because the genotypes of your relatives are correlated with your genotype, this kind of effect is going to show up in a genome-wide association study unless you do something like controlling for parental genotypes. And that could be thought of in some way as a very fine-grained form of population stratification at the level of an individual family. And then if you go to the broader population, there can be other forms of population stratification that are not causal indirect genetic effects, but also contribute to genotype-phenotype association to as Patrick explained before, two subpopulations that are different have different means in their traits, if genetic variants are differentiated between those populations just due to drift, those variants might not have any causal effect. You're going to get some association at that locus, which would contribute to the effect estimation in a genome-wide association study, unless principal components are perfectly adjusting for that, which is possible but probably unlikely. And then another factor that can contribute to the genotype-phenotype association is assortative mating. And assortative mating and population structure are both examples of non-random mating. Often in human genetics, the default assumption is random mating. I'm not sure what that says about the mating habits of human geneticists, that they tend to assume people mate randomly, but both assortative mating and population structure generate correlations between your allele and other alleles in the genome that might be affecting the trait and potentially other environmental factors. So these forms of non-random mating are what are generating these can generate this confounding effect due to correlations induced between that allele and other genetic and environmental factors influencing the trait. And if you use parental genotypes as controls, what you're essentially doing is using the random variation in the offspring genotype due to the segregations that occur during meiosis in the mother and the father to separate out the causal component, the direct effect, as we call it, the causal effect of inheriting a particular allele from these other factors that contribute to genotype-phenotype association in the population. And I should point out that this is very much not a new idea. In fact, this is going back in many ways to the more traditional way that genetics was done in the pre-genome-wide association study era. And one of the more popular tests that was done in that era was called the transmission disequilibrium test. And this was because people were very worried about stratification when they were looking for rare Mendelian disease-causing genes, which can be, looking at rare variants can be very liable to stratification issues. So what people would do is they would take a sample of children who have a particular disease, and they would look at the genotypes of the parents and the genotypes of the offspring. And if you imagine a population of parents that are heterozygous for this particular SNP, so they have an A and a T allele, based on the laws of Mendelian segregation, you expect 50% to inherit the A and 50% to inherit the T. But let's imagine that A is causal for the disease. In a population of offspring that have the disease, you would perhaps observe, if you have sufficient statistical power, a transmission disequilibrium, i.e. over-transmission of the A allele to children that have the disease compared to children that don't have the disease. So you might see in the affected offspring, 60% are inheriting the A and only 40% inheriting the T. And that's very robust evidence that the A allele is actually causing the disease in the child, because the only variation given the parental alleles is just this random segregation. So if you observe over-transmission of the A allele to the affected offspring, it's very robust evidence. So this was a kind of test used in the pre-genome-wide association study era. Then people moved to using large samples of unrelated individuals, which is a lot more powerful to find associations between common SNPs and traits, which was a very successful research program, but it does have the disadvantage of not properly controlling for certain confounding factors. So why should we be doing this now? Why should we go back to this? Well, I think that GWAS has been very successful, but if you want to answer some of the more subtle questions, and especially questions that are relevant, I think, for the social sciences, we need to properly account for the confounding that might be present in what we're doing, and ideally have confounding-free summary statistics that we can use for a lot of the analyses that we want to do. So one thing we can do with this is just assess the degree to which GWAS, standard GWAS, is biased, and we might find that it really isn't biased, in which case we can all go back to doing what we were doing before, perhaps. We can assess the degree to which genetic correlations actually reflecting underlying shared causal effects, i.e. biology, rather than shared confounding effects. In doing this sort of family-based methods, you can get some handle on relationship between genetic and environmental factors affecting a trait. You can create a stratification or bias, indirect effect-free polygenic index, and that could be useful if you're worried about the bias that might be in the polygenic index for whatever application you're thinking of. And these kind of polygenic indices and related methods could be useful for improving prediction within families and also prediction models that take into account genotypes of parents and offspring and family history and things like that. Taking into account this sort of family-based model would be useful. It can help remove bias from Mendelian randomization analyses. I think Dan's going to talk about that later today. And then also studies of selection and other things that are to do with differences between subpopulations. They're very sensitive to the kind of structure, population stratification bias. It might not be relevant for certain questions like detecting a particular allele that's highly associated with a trait, but when it comes to trying to detect selection on a polygenic trait, any kind of subtle bias that's present in the summary statistics can lead to spurious results or inflated estimates, so it's very important for this kind of analysis. So I'm going to go through the derivation of the sort of within, the core kind of within family GWAS or family-based GWAS regression equation in this model that Dan introduced last week. I think I've changed the notation very slightly to distinguish that we're talking about the direct effects of the SNPs here, which we're denoting with delta. So we're saying that this mean-centered phenotype of individual I is the outcome of the causal effects of capital K SNPs, biallelic SNPs, so these genotypes accounting 0, 1, 2 copies of the allele, and I think basically we should probably assume that these are mean-centered for the sake of the notation, and each SNP has this direct causal effect, which is this effect of, you know, if you actually inherit the allele, is it going to change your trait in some causal, through some causal pathway. Then we have these environmental confounds, which, you know, could include various things, including indirect genetic effects. We're going to subsume them into this term. For now, we could write a different model that separates indirect effects from other environmental effects, but we're going to subsume indirect genetic effects from parents and siblings into this term, and then a residual term that's uncorrelated with these. And I'm going to be a little bit sloppy or lazy with the notation for residuals in this talk. So, like, I think basically you can consider the epsilon term to be the residual bit that's uncorrelated with everything to the left of it, because otherwise I'd have to have a different residual for every equation. But it's worth remembering that the residuals are not necessarily the same, but it's a sort of a context-dependent residual, perhaps a way to think about it. So in this model, what's the standard GWAS coefficient picking up? So we're considering GWAS without controlling for any confounds here. Obviously, if you control for principal components, it's going to change things versus not doing it. But let's just imagine we're just simply regressing phenotype onto genotype and getting some regression coefficient. Well, you're going to pick up the causal effect of inheriting the allele that you're counting at that particular SNP K, plus the causal effects of any alleles from SNPs that are correlated with the SNP that you're looking at, and that correlation can be due to various factors that I'll talk about in a second. And then we're also going to be picking up environmental confounding that's due to correlation between the allele, the allele at that SNP and whatever environmental factors. So that's the gene-environment correlation that is the title of the talk. So what are the different sources? So I think that probably the primary source of this kind of confounding is actually local linkage disequilibrium in some sense, but we don't typically think of that as confounding, although it kind of is in terms of really thinking about causality, as in if I change this allele, what effect would it have? Local linkage disequilibrium is confounding in that respect, but if you're talking about identifying a particular genomic locus that has a causal effect, the local LD doesn't matter so much, and we can model it in useful ways. But the more problematic types of genetic confounding are what are the non-random forces of non-random mating that generate non-local linkage disequilibrium. So correlations between SNPs on different chromosomes, for example. And the two primary types, although it's one of these theoretical constructs where we like neatly separate things into assortative mating and population structure, but in the real world, it's not like that. They can't be neatly separated. People are, mating based on proximity, geography, traits, whatever, and it's all a mishmash together in the real world. But in theoretical derivations, you can separate them into pure assortative mating and pure population structure. And both generate correlations between alleles irrespective of their physical position in the genome, which makes it very hard to model and deal with, and can generate this kind of confounding. So, so assortative mating generates. So if we have assortative mating that leads to a correlation between the phenotype of the father and the phenotype of the mother, some correlation RAM, well, that's going to generate a correlation between a SNP that affects the trait at position K in the father and another SNP that affects the trait at position L in the mother. And assuming a linear model, you can derive that the correlation between the genotype of the father at position K and the genotype of the mother at position L is equal to this, and it doesn't rely on the physical position, where it's actually assuming independent segregation there, assuming this is the only source of correlation between the relative genotypes. And this is just what you get from one generation of assortative mating. I think Patrick's going to talk about this in more detail, but it can go on for multiple generations, and it generates these correlations between causal alleles at different positions in the genome, and that's the source of confounding. Another source of genetic confounding is genetically caused differences between subpopulations. So if you were doing, so this is actually showing how you can actually use, although I'm not sure it's the best way of doing it, but people have done this, you can actually use the loadings of SNPs on principal components to infer which SNPs that have been under what's, I think, usually called like a hard selective sweep. So an individual SNP with a really big effect that's been under really strong selection, you can often detect that because it has an outsized coefficient. It makes an outsized contribution to the principal component. So if you were doing a genome-wide association study of lactase tolerance, you might, and you had a mixed sample where, from two subpopulations, one that has been under selection for lactose tolerance and one that hasn't, you're going to find that basically any SNP that's differentiated between those two subpopulations, just because of drift, is going to be correlated with whether you have the lactose persistence allele or not. So that's going to generate this confounding effect basically across the genome because of the genetically caused difference between the subpopulations. And anything, this is showing it for these simple kind of classical hard sweeps where you have a single locus that's really been selected for and generating a difference between subpopulations. But if you have a more polygenic trait, you could have something similar to this, but it's in a more subtle way. Then population structure, yeah.
So what we saw in the Manhattan plot is how much the SNP is, how significantly the SNP is associated with PC.
So this is a slightly more complicated thing than that. It's basically, I can't remember the details of this. It's from a while back. I remember reading it, but they basically have a way of figuring out whether a SNP is has a bigger loading than you'd expect based on drift. So this p-value is the p-value from some test statistic to say whether the loading on the PC is bigger than you'd expect based on a model of drift. Then there's, population structure can be correlated with environmental effects, and that's how you get environmental confounding. And even in a relatively homogeneous population, such as the UK, there are clear and distinct genetic clusters that are in different parts of the country that have different environments. This is from this was work done in my group during my PhD. I wasn't actually involved in it myself, but they're using this method called fine structure that's actually a lot more sophisticated than just using principal components. It uses haplotype-based method to infer clusters, genetic clusters, in the population, and you can see there are all these different clusters identified even in the UK. And these clusters might not necessarily be identified particularly well based simply on principal component analysis. That's the argument they make anyway, that these haplotype-based methods are actually much better at detecting the genetic structure in the population. And this I'm showing here as a point of comparison is from this paper by Abdelouai et al. In 2019. They got a lot of media attention, and in that they show this spatial distribution of the educational attainment polygenic index in the UK Biobank, and they make an argument. So they highlight, so I'm from here, which is depleted for, no, this is the northeast of England, so where part of the early industrial revolution, coal mining and whatever all came from the northeast of England, railways and stuff. So their argument is, these regions have been depleted of people with high education polygenic indices because of economic-related migration, which is an argument that makes some sense. However, it's hard to really distinguish the true signal from the stratification, in my opinion. They adjust for 100 principal components, but as I was saying, these principal components are just linear functions of the genotype matrix. They're not doing anything that sophisticated like this method is. And one thing I noticed when I put these next to each other is these regions that they highlight as coal mining regions seem to be regions that particularly have their own genetic cluster. So what they might be picking up, and also they seem to be saying people in the Highlands of Scotland, they're really the smart ones, so they're really the geniuses of the UK. So I have some skepticism that they're all of, well, part of my family's from the Highlands too, to be fair, but I have some skepticism that all of what they're observing here is real genetic signal. I'm sure there's some real genetic signal, to be honest, because there is a lot of economic-related migration from more deprived, like post-industrial regions. I'm sure it's a similar pattern in America. There's some real signal, probably, but is it all real signal, or is some of it confounding? I'm not sure we can be totally sure, but maybe if we use the direct genetic effects instead of, there might be less confounding in an analysis like this. And part of the reason I'm skeptical is because principal components, especially those based on common variance, which is basically what everyone does in GWAS, they're actually not very good at capturing recent structure. And this goes back to the population genetics lecture that James was giving yesterday. We have these coalescent trees, and depending on where you drop that mutation, how far back in the tree you drop that mutation, it's going to determine different points in population differentiation. And common variants tend to be very old. Most of the common variants in non-African populations probably derive, I think I remember reading, and they derive from near the bottleneck which happened during the sort of out of Africa dispersal. So a lot of these common variants that are used to construct PCs, they're picking up really old structure in the population. And they're not picking up recent structure. So this is from a recent paper that I think explains all this very well, showing that in a simulation where the structure is only from the last 100 generations, principal components based on common variants don't really differentiate the different subpopulations in this model of population structure. If the structure is really old, they do a good job, although still not as good as PCs based on rare variants. So these are really rare. These only have a minor allele count of 2, 3, 4. So they're only present in a few people. And because they're only present in a few people, they're telling you about their more recent mutations that are telling you about the differentiation in the population that's happened in the more recent generations. So I think that when it comes to stuff like the structure in the UK population, that's probably within the last, a lot of that is generated more recently, and maybe the principal components everyone's using are not perfectly adjusting for that kind of structure. They're doing something, but they're not doing everything that we'd want. Maybe using rare variant PCs is an interesting avenue to go to, but the problem with that is you need high quality genotypes of rare variants, which you're only going to get with whole genome sequence data in general, at least for genome-wide. If you want genome-wide ones, maybe exome, you can get the exome or something more easily. So that's all the issues that we might hope to solve. And how do we do it? So we basically exploit the statistical consequences of meiosis, and nature's been very kind to us. And it's basically created all these little natural experiments or randomized controlled trials or whatever for the effect of genetic variants on traits. And I just should probably spend a second explaining the notation here. So this is the individual I's paternally inherited allele at position K. And so what allele they inherit is the outcome of the meiosis. They produce the sperm in their father, and what they inherit, whether they inherit the father's paternally inherited allele or the father's maternally inherited allele, that's the outcome of this meiosis segregation event, which in general is just like a random coin toss that's going to be independent of all the confounds. And similarly, the individual's maternally inherited allele is randomly either the mother's paternally inherited allele or the mother's maternally inherited allele. So we're all sort of these mosaics of our four grandparents' alleles, and which of the paternal grandparents we get is random, and which of the maternal grandparents we get is random. And basically, if we condition on the parental alleles, then the variation left in the offspring genotype is random and unrelated to confounds. So we can say that the genotype of the individual is conditionally independent of the confounds given the parents' alleles. And that's how we get to remove the confounding when we do a family-based GWAS. And based on that idea, we can split an individual's genotype into a within-family component and a between-family component, where the within-family component is due to the segregations in meiosis, and the between-family component is the expectation of the individual's genotype given the parents' alleles, which is actually just the average of the father and the mother's genotype. And then that allows us to write the individual's genotype as this average of the parents' genotypes plus this segregation. I'm just going to call it a segregation genotype. So that's the within-family variation. And these two components are independent of each other. And this is shared between siblings, and this is different between siblings. That's why I call it within-family variation. And this varies between families, but not within families, basically. So these are sort of orthogonal components of the individual's genotype. So we can ask how much variation is in each component, and I think this is often a surprise to people who haven't seen the result before. But we're assuming random mating here. The variance of the individual's genotype is 2f1 minus f, and the variance of the average parental genotype is just f1 minus f. So averaging reduces variation. So then, just by subtraction, the variance of the segregation genotype is f1 minus f. And what that implies is half of the genetic variation is within families. which is kind of a surprising result when, or at least I found it surprising when I first thought about it, because you kind of don't think that half of the genetic variation in the population is actually within siblings, basically. Siblings can capture half the variation. Important caveat, though, is this is assuming random mating. And if you have, at the level of an individual SNP in a relatively homogeneous population, random mating is usually a very good approximation. There are obviously going to be some SNPs that don't follow that have something strange going on. But for most SNPs in a relatively homogeneous population, like UKB white British or whatever, I know that's a controversial name or whatever, but like some sort of homogeneous relatively homogeneous populations is a good approximation. If you looked globally, more variation would be between families than within families because of the structure in the population. And at the level of polygenic indices, things can be a little bit more complicated. When you have assortative mating is going to push down the within-family variation and increase the between-family variation. But at the level of individual SNP in a relatively homogeneous population, it's a good approximation.
Excuse me.
Yeah.
What is F?
Oh, sorry. F is the frequency of the allele that you're counting. So for the genotype, you're counting 0, 1, 2 copies of the allele, and F is just the frequency of that allele that you're counting. So then F1 minus F is commonly called the heterozygosity. Oh.
Okay. And do we know what is inside the within family, the delta?
Well, that's just the deviations around the expectation given the parents. So it depends on, yeah, I mean, it depends a bit on what the parents' alleles are. Like if the parents are both homozygous, then there's actually no variation. So this is, you know, the average over, you know, the possible parental alleles. So it depends a bit on what the parental alleles are. If parents are both heterozygous, then the average is the expectation. Well, it depends on the population frequency as well. So it's basically just the deviations around this average. So if you take the individual's genotype and you subtract the average of the parents, that's what that is. So it can be positive or negative, I should point out. And it has expectation 0. So then to get towards the actual within-family GWAS equation, you can define the parental genotype to be the sum of the father and the mother's genotype. So that can go from 0 to 4, although we're assuming mean normalization here probably, just for notational convenience. But if we weren't mean normalizing, it would go from 0 to 4. Then if we imagine that we have the parental genotype and we have the offspring genotype, we can compute this segregation genotype just by subtracting 1/2 or the average parental genotype from the offspring genotype. So you can actually compute this fairly trivially if you have the parental genotype. And then based on this relationship, you can rewrite the model that I introduced before in terms of the segregation genotype and the parental genotype. So you see here that the segregation genotype is picking up the direct effect, but actually the parental genotype is picking up 1/2 of the direct effect too. And what that's telling you is direct genetic effects are contributing both to within-family differences and between-family differences, which makes sense if you think about it, really. So then we have these properties that the segregation genotype at SNP K is independent of the parental genotype at SNP L for all SNPs L. That's sort of by construction. It's independent for SNP K and also for all the other SNPs. So if we want to regress phenotype on the segregation genotype, then we can ignore this whole parental genotype term. And we also have from before that the segregation genotype is independent of the confounds, because that's the component that's just due to this random segregations in the family, which are uncorrelated with the confounds. So we can also ignore the confounding term, and we can ignore the residual term. So if we're regressing phenotype on the segregation genotype, it's only this term that contributes, and therefore we're going to pick up the causal effect of that SNP And then we might wonder, well, are we picking up the causal effects of other SNPs due to linkage disequilibrium? And the answer is yes, but not as much as in standard GWAS. So basically, the segregation genotype at SNP-K is independent of the segregation genotype at SNP-L if SNP-L and SNP-K are on different chromosomes. And this is because chromosomes segregate independently within a family. So you're not picking up, there's no correlation, essentially, for this within family segregation, this deviation around the expectation given the parents across chromosomes. There's no correlation.
Assuming random mating.
No, this is not assuming random mating. This is true whatever the parent, the non-random mating affects the correlations in the parents' alleles, but given the parents' alleles, meiosis is happening, chromosomes are segregating independently and it's breaking those correlations across chromosomes. So that's why we can say, regressing on the segregation genotype, we're getting the causal effect of that individual SNP, and then we're picking up some fraction of the causal effects of other SNPs on the same chromosome in proportion to their LD, the LD due to physical linkage. So this is actually kind of the assumption that LD score regression a lot of methods make, which is true for this kind of within-family regression, but it's not actually true for standard GWAS. And basically, the only things you're picking up are the things that are in local LD due to physical linkage in the same chromosome. But for the sake of notational sanity and convenience, we're going to just assume that causal SNPs are on different chromosomes, so we're going to ignore this term. It's not really true, but we're just going to ignore this term for the sake of the derivations here. And actually, this kind of assumption, while false, is often useful theoretically. And if you think about it, most pairs of SNPs are actually on different chromosomes, so there's some logic to it, but it's obviously untrue in reality. So because the segregation bit and the parental bit are independent, we can just consider doing two separate independent regressions to figure out what we're picking up by doing a family-based GWAS with controls for parental genotypes. So now we're going to regress the individual's phenotype onto their parent's genotype, and that parent's genotype is the sum of the maternal and paternal genotypes. And we're going to pick up 1/2 of the causal effect of that SNP, and that's because you have this term here. So the direct effect is contributing to this sort of between family component. And we're also going to pick up, so if we stick with this unrealistic but perhaps useful assumption that causal SNPs are in different chromosomes, we're going to pick up genetic confounding. And this, let's just say this is correlations between SNPs on different chromosomes induced by non-random mating, population structure, sortative mating, selection, whatever, is inducing these correlations between SNPs that would be independently segregating within a family, but at the level of the parents, they're correlated because of these forces of non-random mating. So you're picking up this genetic confounding term, and you're also picking up environmental confounding terms. So the correlation between the genotype and the environmental factors going through the combined parental genotype. And we're going to subsume, so this would be including indirect genetic effects if they exist. So we're going to subsume these terms into this alpha coefficient. So then the regression on the parental genotype gives you 1/2 of the direct effect plus this confounding term. So then we can basically combine the results of these two independent regressions to derive the within-family GWAS regression equation. So we have the expectation of the phenotype given the segregation genotype is just the direct effect of that SNP times the segregation genotype. Expectation of the phenotype given the parental genotype is 1/2 of the direct effect plus the confounding effect times the parental genotype. But we can combine this 1/2 of the direct effect times the parental genotype with the segregation genotype, and putting those back together, we get the original individual's genotype. So then, by combining these two together, we get the individual's phenotype is the direct can be written, at least, as the direct effect of this SNP, and then this alpha term times the parental genotype, where this alpha term is picking up all of this. And we're going to call this alpha term the non-transmitted coefficient. It's kind of, you might think we could have a nicer name, and I tried that, but I think that an issue with calling it something parental kind of implies that it's picking up some sort of causal effect of parents and offspring, and that's not necessarily the case. This alpha coefficient can be picking up pure confounding. And why do I call it a non-transmitted coefficient? So there's another way of writing it, of this equation. So the parental genotype, there should be a K there. The parental genotype is comprised of 4 alleles. 2 of them are transmitted to the offspring, so that's the offspring genotype. 2 of which are not transmitted to the offspring, that's the non-transmitted parental genotype. So we can write the parental genotype as the sum of the alleles transmitted and the alleles not transmitted to the offspring. And then that allows us to rewrite this equation in terms of the non-transmitted parental alleles, and this alpha thing is the coefficient on the non-transmitted parental alleles, and that can be used in some sense as like a control to estimate the direct effect of the SNP. And I should point out here, this epsilon, I put a K here, because I wasn't being lazy this time, to differentiate this epsilon from the original epsilon. The original epsilon was the residual accounting for all the direct effects of all the SNPs and the confounds. This epsilon is just the component in the phenotype that's uncorrelated with the individual and their parents' genotypes at that SNP. So this is including all the other SNPs in the genome, the effects of all the other SNPs that are not correlated, captured by this SNP. Yeah?
Could you explain again how you go from the 2 equations at the top to the within family regression equation?
Well, they're 2 independent regressions. So the joint regression is just the sum of these 2 things. And then if you have a joint regression, then the residual is uncorrelated with whatever you've done the joint regression on.
So this is the within family regression equation is a regression of the outcome on the individual's own genotype and the parental genotype.
So yes, it's equivalent to a joint regression on the individual's genotype and the parent's genotype, and I've sort of just derived what these are in terms of the other model. You're adding the right-hand side? So I'm just adding these together, and then if you combine this term with the delta k bit of this term, you get the individual's genotype back, and then you have the alpha k times the parental genotype, and then because you're adding 2 independent regressions together, it's equivalent to a joint regression on both of these. The residual term is just whatever is uncorrelated with those 2 terms.
I think the only tricky step is that the x is equal to delta plus x par over 2.
Yeah.
And so when you factor out the delta, that's exactly what you're left with.
Got it, yeah.
Yeah, it's not like a super rigorous way of doing it, but it's a straightforward way of doing it by splitting it into 2 independent regressions and then combining. And then from this equation, you can derive what standard GWAS, assuming random mating, is estimating in terms of these parameters, and it's basically estimating the sum, and that's basically the same as what I showed in the earlier slide. And we're going to call this the population effect, which is just like the sum of the direct effect and the non-transmitted coefficient. So I hope I've convinced you that doing within family GWAS is like a sensible thing to do if you want to have an estimate of something that you understand what it is. But I guess the question is why isn't that what people do in general? And the main question is, the main reason is power. So the power of a within family GWAS is much smaller than the power of a standard GWAS. And the main reason for that, really, the primary drop in power is because you have to restrict your sample to the subset where you have parental, at least the way I've presented it here. I'm going to talk about different ways in a sec. But the way I presented it here, you have to restrict your sample to people with both parents' genotypes. And that's going to be a pretty small subset of your data in general. So that's your big loss in power. And then even within that subsample, you're only using the within family variation to estimate the direct effects. That's 1/2 of the genetic variation you're using in standard GWAS. So even within that subset, the standard GWAS is going to have twice the effective sample size, or your variance for the direct effects is going to be approximately twice that of the variance for the population effect that you're estimating in standard GWAS. And obviously with standard GWAS, you can use a huge sample of unrelated individuals as well. So you have a lot more power to discover genotype-phenotype associations, estimate things. It's just that it's a bias versus variance trade-off thing in some sense. Yeah?
So I'm just making sure that I'm doing the math in my head right. So if there is assortative mating, the power would actually be even worse than, you'd need even more than double the participants.
Yeah, although for polygenic traits, assortative mating has a negligible influence on the individual SNP within family variance versus between family variance. At the level of a polygenic index is going to have a much bigger influence, but at the level of an individual SNP for a polygenic trait, the effect is negligible.
So that's, I mean, this would be a diversion, but it might be an interesting thing to talk about how that revelation is what led to I don't know if it was a retraction, but to a reinterpretation of some findings from on social homogamy. Are you familiar with this? I think it was Ben Domingue and Dalton Conley had found that people who were friends were more genetically similar to one another, using the genomic relatedness that they use in Gremel. And the thought was that they were assorting on traits that were polygenic, and that was inducing relatedness. But there was a commentary, I think it was by Adel Abdelawi, that basically, I mean, it's not something I had thought about. It's a very important point, which is that when you assort on traits, you're not assorting on SNPs, you're just assorting on the cumulative effects of many SNPs.
Yeah.
So it doesn't mean that people are going to be concordant on the individual loci. They're going to be concordant on the y hat, so the polygenic score, which means that there's, just as you said, that the assortment at the level of the SNP is pretty minuscule when you're sorting based on traits as opposed to based on individual genotypes.
Yeah, I mean,
at the level of the locus.
Unless like all SNPs were causal for your trait or something, then I guess relatedness. Well, but I guess it's different, isn't it? Because it's just the value. There are many different ways to get to the same trait value with a yeah.
Right? So like, if 10,000 are causal, but a person who's high only has 500 of the trait increasing variants, it could be 500, any of the 500 out of 10,000 that are causal, and someone else that they are friends with or assort with romantically could also be high for genetic reasons, but on a completely different set of 500 out of the 10,000.
Yeah, I mean, I guess if what they were claiming was true, we'd expect spouses to be like significantly related to each other.
Yeah, they'd be co-sanguis, yeah.
Yeah. Well, I guess they are in some places. So. Okay. So I presented things derived in terms of parental genotypes, and I think that's the sort of right way to think about it, at least. But in practice, what has often been done is using sibling genotypes instead, genetic differences between siblings. So I just got a slight change in notation here. So this would be the sibling 1 in family I, and I guess I should say family is not to mean anything other than individuals that share the same set of biological parents. And what you have here is, I said, genotype is sibling 1 in family I, and then the parental genotype in family I, and then for sibling 2, they're going to share the same parental genotype, because that's how I've defined the, full siblings share the same set of biological parents. So then if we take difference between the siblings' phenotypes, and we get the difference, the direct effect is then going to be, the difference between siblings due to direct genetic effects is going to be due to the genetic differences between the siblings. And, because the average parental component of the siblings' genotypes is the same, that cancels out. So you're really just using the segregation genotypes in this estimation of the direct effect. And you might hope that this is uncorrelated with this. Actually, it's not quite if you have indirect effects from siblings, which I'll talk about in a sec, but let's just assume that this is true. Then by regressing phenotypic differences onto genetic differences in siblings, you get an estimate of the direct genetic effect. And what's the power of that? Well, the power of that depends on the correlation between the siblings' residuals. And if you're doing an individual SNP for a polygenic trait, that's basically the phenotypic correlation of the siblings. So the more correlated siblings are, the more powerful this method is actually, because if you take the difference, then the more correlated the things are, then the lower the variance of the difference. So this looks okay relative to what we had before, but I would point out that this is the number of pairs, not the number of individuals, the number of SIB pairs. So there would be 2 n individuals, n pairs. But if you had this sample of sibling pairs, but you actually also had the parental genotypes, and you use the parental genotypes as controls instead of the sibling difference approach, this would be your variance for the estimate of the direct effects. And actually, this is up to the variance from the sibling difference thing is up to twice as large as the variance from using the parental genotypes. So in general, the parental genotypes are giving you a lot more power than sibling differences.
So if you have half siblings here, then you don't use them.
So half siblings, because they don't share both parents, they can't be used in quite the same way to get unbiased estimates of direct effects. If you have parent of origin and phasing and stuff, there's a way you could use them, but it's not as simple as it's just taking the difference. So what I said before is that this has an issue if you have indirect genetic effects from siblings. So I've been ignoring indirect genetic effects from siblings before, because when you're deriving things in terms of parental genotypes, you can ignore it safely and subsume it into this alpha term. But when you're doing genetic differences between siblings, this is actually a case where indirect genetic effects between siblings violate the assumption. Well, it's not quite violating the assumption, but because you're now using a pair of individuals, the residuals from before, even though they're uncorrelated with the individual siblings' genotypes, the residual can be correlated with the other siblings' genotype when you have an indirect effect from siblings. So when you have this indirect effect from siblings, which I'm denoting here, so the individual sibling 1's trait depends not only on their own genotype, but the siblings' genotype through the environment. And you have to change the coefficient here in the parents, because now you're taking this out, and before the parental coefficient was capturing 1/2 of the indirect effect from a sibling. Somewhat technical diversion to go into all of that. But basically what matters is if you take the differences of the siblings' phenotypes, you're actually estimating the direct effect minus the indirect effect from the sibling. So there can be this confounding with the sibling difference approach if you have indirect effects from siblings. It's not going to have confounding due to population stratification or anything like that, so it's still sort of a causal effect estimate, but it's sort of like a linear transformation of these two causal things. It's not exactly what you'd want in an ideal situation. Yeah?
I did a bad job explaining this in my original talk. So can you give an intuition for why this sibling indirect effect shows up when you do the sibling fixed effects regression?
Well, because if you're looking at genetic differences between siblings, then both the indirect effect from siblings is making them more similar, and the direct effect can explain the differences. So you get this difference between the direct effect and the indirect effect, explaining the effect of the genetic differences on the phenotypic differences. They're operating in opposite directions, essentially. They're both kind of, well, perhaps the way to think about it is they're both determined in part by the segregation events. So what genotype the sibling inherits is determined by segregation events in that sibling. What genotype you inherit is determined by the segregation events in the different segregation events leading to your genotype. But when you have indirect effects from siblings, both segregation events are playing into your trait. So you need to consider both if you're going to look at this difference between them.
So the way you've written it down here, that if your sibling has a positive indirect effect on you, it is positive. That means that when your sibling's genotype is the difference when the difference between genotypes is bigger than for any given difference in genotypes, because of that indirect effect, the difference in phenotypes is smaller, and that's why the gain is coming in negative. That's a way to.
Yeah, if you just imagine like these are the two, the outcomes for the siblings, and your genotype increases, then your genotypic, that's going to increase your outcome a little bit, but it's also going to increase your sibling's outcome a little bit. And so, you're seeing your own increase, but that gap's closing because of the increase in your sibling.
Yeah, they're operating in opposite directions, and I guess the thing to realize actually in why this is not independent of this when you have indirect effects is the assumption here is that this epsilon I1 for sibling 1 is uncorrelated, independent of the individual's genotype and the parent's genotype. But actually, this epsilon I1, if you have indirect effects from sibling, is including part of the indirect effect from the sibling, from sibling 2. And this epsilon I2 is including part of the indirect effect from sibling 1. So the difference between them has got this indirect effect component, which is not independent of this difference.
So something that they talked about from time to time in the twin literature, that it's just something that you said kind of reminds me of it, which is that you're expecting that the indirect effects from the siblings to basically in the same direction as the I guess the direct effect. In the old twin literature, they would talk about assimilation and contrast effects.
Yeah.
So sometimes if your sibling is the athlete, then you go to great lengths not to be the athlete.
Yeah.
Even if you're just as built as him or her, right? So I could see. I mean, the math will still.
They're pushing in opposite directions, I guess, still in this difference comparison.
Right. I would expect it still to confound things, but not necessarily in the way that you described. Isn't it just
that would just
positive or negative?
Yeah, it would be a negative beta.
Yeah.
Sure. No, I know. It's just that the language you were using was. Was. I'm not saying that the math is incorrect. I'm just saying the language you're using it was implicitly assuming that the indirect effect from the sibling is going to be in this kind of concordant direction, and I just think it's if you get back to the substance of the social sciences and you think about what sibling effects are, in some cases for some traits in some contexts we might actually expect contrast effects as opposed to assimilation effects, which I just think is interesting and maybe of substantive interest to
people were running some of the model.
Yeah, no, that is a very fair point. That these can be pushing similarity or difference between siblings. Okay. So, given the caveats that maybe the sibling differencing is not the ideal thing to do, it is actually a fairly straightforward thing to do. And there's a lot of sibling genotype data available. So there's this recent meta-analysis of using the sibling differences to estimate the within family. They call it within family effect, or it's a sort of an estimate of the direct effect, but maybe with a little bit of bias from the sibling indirect effect, but probably nothing. Well, we don't really know how. It's still a matter of debate whether these things are substantial, and I guess what direction they go in as well. So, but nevertheless, this has been done for quite a large sample in this preprint on bioRxiv, and this is one of the figures showing that comparing basically the size of the estimate that you get of the direct effect from the sibling difference approach to the estimate of the population effect. And the way that this plot is, the further something is from this 0 point, the more the direct effect is shrunk relative to the population effect. And that shrinkage could be the outcome of various forces. Yeah?
So if there's something like with 2 siblings and something with 3, 5, or 7, how does this account for the differences in the slip size?
So I think the way that they did it, it's a good question. So this doesn't really generalize very well to multiple siblings. And I think the way that they did it is that you can basically subtract the mean genotype among the siblings from the genotype of the individual, and you can regress on that. And that's kind of very similar in some sense to subtracting the average parental genotype. And in fact, as the number of siblings goes to infinity, the average among the siblings would go to the average parental genotype. So it would become the same thing as the family size goes to infinity. So that's one way of doing it, that you regress on the deviation of the individual sib from the average of the siblings' genotypes. That's the way to generalize it to multiple siblings, but then you have to do some more modeling of correlations between the siblings in the regression, whereas this kind of accounts. If you just have sibling pairs, this is quite an elegant way to do it because you create a set of n independent families, so to speak.
If you did that mean genotype trick, then would this beta, the eta bias be like the average indirect effect among your siblings, or how would that look? It's not quite that simple.
Yeah, I think it would be different. I'm not sure exactly. I have to do the math, but I mean, the thing is, when you get to multiple siblings, like the indirect effect from the sibling could be completely different when you have, depending on family size as well, right? So what the appropriate model is in general for these indirect effects from siblings can be quite complicated. Then there's also this. They used LD score regression to estimate SNP heritability using the direct effects estimated from the sibling difference, and they compared this to estimating SNP heritability using the standard sort of GWAS coefficient from the same sample. And I'll have more to say about the variance components and heritability later, but just pointing out here you get very different estimates for education and for height than if you use the within family coefficients versus the standard GWAS coefficients. So there's clearly some difference going on in what's being picked up. Is it heritable variation, sort of mating? There's all sorts of reasons why these things can be different, but they are actually quite dramatically different, for example, for educational attainment.
Alex,
Yeah.
Might be a good time to just take some questions and then take a break.
Oh, okay, sure. Okay, do people have questions? Yeah.
So I was wondering, like, how the models are dependent on the type of phenotype that we're looking at. So if a phenotype is prone to within regular GWAS versus
so I think the models are probably not so dependent on the phenotype. I mean, you might make different modeling choices depending on the phenotype. If you have a good reason to believe sibling indirect effects are not important, then ignoring them can actually gain you a lot of power. However, the sort of general theoretical modeling doesn't really depend on phenotype. I guess if you have a non-linear, if you have a binary outcome or something, then things can maybe be more complicated in theory, or other kinds of non-linearities going on. I've been assuming an underlying linear model here, so that might change depending on the phenotype. I think the results that you get do obviously depend a lot on phenotype, and you're correct, the educational attainment does stand out, and traits related to educational attainment do stand out as showing large differences between whether you do the within family thing and or the standard GWAS thing. I think other traits as well. I mean, there's height. This is LDSC, but the shrinkage thing is a lot of shrinkage. Height. That's educational? Is that BMI?
That's education.
That's education. It's hard to tell from here. Ever smoking, age of first birth. So, and that similar to results we've got from other methods and other data sets, that like things related to behavioral traits, fertility related traits, education and cognition related traits tend to show the biggest differences. Things that are a bit more like classically sort of biological probably show less difference, although, you know, a priori I'm not sure we really know in advance. Like HDL, actually, you know, that's high density lipoprotein cholesterol. That often shows a bit of a difference between these things. And so, I mean, things that are like classical biomarkers of disease can also show these differences, but you probably, I would guess that the biggest differences you're going to see are probably for the more behavioral traits. Yeah.
I noticed earlier you were accounting for within and between family variation, and here in the case of siblings, I've noticed that you've been looking at within sibling pair variation, but what about between sibling variation?
So, so you mean, what exactly do you mean by that? Because within sibling, within an individual, there's no variation. Between two siblings, the variation between siblings is the within family variation, and then the variation between families is shared between siblings. So that would be the expected genotype. So the siblings have an expected genotype given the genotype of their parents, and that's what causes between family variation. And then the variation around that expectation given the parents' genotypes is the within family variation. I don't know if is that what you meant?
Yeah, I guess I was just thinking about most of the differences between sibling pairs versus the sibling pairs.
So you mean different kinds of sibling pairs? Yeah, so I think Dalton showed some results implying that there was an indirect effect between female twin DZ twins and not between male DZ twins. So certainly there's no reason to suppose that these things are the same across different kinds of sibling pairs, and probably differences in age, in sex, in other factors can influence these things if they exist. It's possible to fit models that allow you to detect those differences between different kinds of sibling pairs, but whether we have a lot of power, certainly at the individual SNP level, there's limited power for that kind of thing. At the level of polygenic index analysis, there's probably more power. Yeah.
What would you say is the meaning of these differences that we observe between the within family estimates and the population estimates in terms of practical utility of polygenic scores? Because the SNP heritabilities differ dramatically, but the effects, the betas, are kind of the same.
Well,
for educational attainment.
Well, I'll have some more results that pertain to this later, but it is, so I guess do you mean what are the practical consequences, or what are the causes of it?
What are your thoughts on the consequences of the different estimates?
Well, again, I think it very much depends on what the goal of any analysis you're doing is. I mean, for certain analyses, the differences are not important. And I mean, it might also depend on what the cause of the differences are. Like, for height, probably most of the difference is caused by assortative mating. And that is probably a less problematic difference, because that will generate less in terms of bias. But it can mean that the standard GWAS coefficient is inflated, but they're still basically the same. One's just like a scale factor version of the other. So the inflation due to assortative mating of the marginal GWAS coefficient, does it matter that much? I mean, it depends. If you want to measure heritability, it matters. But is it going to be a major source of bias in like GWAS? Probably not. It's probably not a major issue. However, even for height, where there probably isn't as much stratification as there is for a trait like education, even for height, there have been issues in doing these studies of selection and things like that due to residual stratification. So even subtle, small amounts of stratification in the GWAS summary statistics can cause your inferences to be highly inflated or incorrect for certain kinds of analyses, like Mendelian randomization or studies of selection. So I think the answer to that question is quite complicated. It depends both on what the causes of the differences are and on what analysis you want to do, like whether your analysis is particularly sensitive or not to the issues that might be present in the standard GWAS summary statistics.
What might be the cause of difference that gives you a similar beta estimate but a different heritability estimate, as it happens with height and education?
So I don't think the beta estimate is that similar for education.
What are the two dots?
It looks
correspond to?
So I think the figure is misaligned somehow compared to the.
Well, I think the confusion is that the black dot and the orange dot are close to each other.
Yeah. Oh right. So
the difference is deviation from the.
Do you see the vertical line? So education, I find it really hard to read from there. So education shrinkage is like 50%. So that's pretty big.
Because it's very far from the vertical line.
And height is like 10% or something. So and shrinkage I think is not really the best statistic anyway for things. So what's the orange and the blue? I can't remember. I don't think it really matters. Yeah.
The lesson is there's real value in investing in good figure design.
Yeah, it's not the best designed figure. It's kind of confusing.
So I think that just at a very kind of moving out to the big picture at a conceptual level, sometimes I get frustrated when people talk about direct effects as estimated using methods like within sibling or within family GWAS, and say that those are the ones that actually occur through biology. In some sense they do, but like I think we need to keep in mind that a direct effect as estimated from within family GWAS of, for example, nicotine receptor on lung cancer is still fairly indirect.
Yeah.
Right? It's working through people having a tendency to smoke more cigarettes.
Yeah.
And so I think it's just important that we don't, I mean, people do this with heritability as well. We don't conflate our lay definitions of these terms with the technical definitions.
Yeah. Yeah, no, that is an important point, and I think the terminology is potentially misleading in that respect. I know Dan agrees with that. And we did think about trying to use different terminology, but I mean, it's maybe something we could talk about. I feel like this whole area is kind of in a bit of a terminological crisis. and probably needs someone to write a paper defining defining all these terms and giving them appropriate interpretations. And I agree that direct effect is misleading. It makes you think, oh, it's this really like biological, like, you know, straightforward thing, and it's not. So yeah, I think it's a good point.
Yeah, I mean, I think even just mentioning the caveat is at least for a sophisticated audience, is all that's necessary.
Yeah.
Sort of a conceptual question. So when you motivated the sibling and family GWAS models by talking about the SNPs that the parents have and the children inherit and the probability of LDs. But essentially what we're doing here, and I may be wrong here, but if we're just controlling for anything that's shared, it's very much similar to like kind of the case model where we're taking out anything that's shared between the individuals. So it could just be, it's just controlling for the environment.
It shares some similarities to the ACE model. I would say it's different in that it doesn't make, the ACE model requires an equal environment assumption. You have to assume that the environmental similarity of MZ and DZ twins is the same if you want to claim that your estimates of the heritability are unbiased in the shared environment. This does not make that assumption. It makes an assumption that Mendelian segregation during meiosis is independent of confounds and independent between chromosomes, which is about as secure assumption as you get in biology. So I would say it's similar to the ACE model in that you're using this kind of within family variation, which the ACE model is also using. It's using the difference between MZ and DZ twins, which is kind of like a within family thing to some degree, but it's not making one potentially problematic assumption that the ACE model makes about the equal environments.
So building on that, if we wanted to be a little less restrictive to be able to use more data, this is a question from Dan earlier for a second. If we were to control for parental phenotype in the case of education particularly, we will be controlling loosely for somewhat for parental phenotype, also for environment.
Yeah. So I think controlling for parental phenotypes can certainly remove confounding. However, I think that there's no guarantee that, I guess it's one of those questions about what's the purpose of your analysis. So that might be a good thing to do for certain things. But if you want to remove all bias from indirect effects and population stratification, etc., it's not going to do that unless you make strong assumptions about what the nature of the confounding is. Also, you know, in a way, I think parental genotypes might actually be more reliable and available than parental phenotypes in many cases. So I think it's potentially a way that should be explored perhaps more than it has been, controlling for parental phenotypes, and but I don't think it's comparable.
I mean, it seems like, yeah, I mean, if your choice is between controlling for nothing and controlling for environmental stuff that you think may be confounding, like, perhaps that's better than controlling for nothing at all, if you're really depending on the things being, you know, somewhat more causal.
Yeah.
But you know, yeah, obviously controlling for the parental genes is going to be better than controlling for environmental variables, you know, which because if you control the environmental variables, you have to assume you're controlling for all of them and in the right functional form. So you're kind of putting your finger in the dike, sort of a thing. Whereas controlling parental genes just solves the problem.
I guess one potential bonus of controlling for parental phenotypes, as long as they're not phenotypes that could be influenced by the child, so I guess that could have an issue. But you could increase your power, actually, as well, potentially, if there are things that explain a lot of variation.
The other issue is that, we gave the example in the PGI lecture about how if you're doing a PGI analysis and you control for the parental education, this was exactly the example for the Papa George and Tom. Because there's error in your PGI, it's possible that controlling for the parental phenotype is actually going to bias the association in the PGI by absorbing some of the genetic effect, right? And so that's another consideration that you need to worry about if you're controlling phenotypes rather than directly with the genes.
Yeah.
Like ideally, if you want to get rid of the indirect effects of siblings, you would run a sibling GWAS on siblings raised together, and then raised apart, and then get the effect of right? Like.
So you're saying if you want to actually estimate the indirect effects.
Well, if you want to get the pure genital effect.
The causal sort of. And yeah. I mean, that I think is a tricky. I mean, that is a study design that could potentially work, and I guess that's related to. Some people have done related stuff where they've looked at differences between like adopted and non-adopted individuals.
So you have to assume that those groups are comparable.
Yeah.
Right? Like what the sorts of things that make a pair of siblings to be reared apart, makes them really different than a pair of siblings that are raised together. Right? It's tricky.
But yeah, I think one thing you can do that is something we're doing at the moment is you can extend this model where you're using the random segregation another generation back to the grandparents, and then you should be able to then use the segregation of the parents' genotype around their grandparents to estimate the indirect effects, get an unbiased estimate of the indirect effect. But obviously that puts a lot of requirements on the data that make it hard to get such samples.
So we should take a break now. This is really important stuff, so we're going to continue shortly. Let's take a 10-minute break and continue at 10 minutes after 11.